Good morning. My name is Professor Evelyne Jacqz. I am working in Hôpitaux Robert Debré in Paris as pediatric pharmacologist. I want first to thank the organizers for selecting the importance of translational research in developmental pharmacology to facilitate pediatric clinical drug evaluation for an oral presentation. This presentation was prepared with EPTRI partners, having the following objectives: to summarize how to develop safe and effective dose in different pediatric age group and different pediatric diseases based on preclinical data. Where objectives were: 1, to develop new knowledge in developmental pharmacology; 2, to validate pharmacological tools to be also translated to the clinical phases of development; 3, integrate data from non-clinical studies and promote innovation to conduct the early clinical phases of drug development in children; 4, increase research of key developmental areas through a specific centralized platform on developmental pharmacology, as presented on the right of the slide; 5, develop and diffuse expertise to all professionals involved in pediatric drug research; and 6, train all researchers and health professionals. First, there is a need to increase knowledge in developmental pharmacology to better understand the factors that impact the pharmacokinetic phases of drug disposition, presented on the left in the cartoon by Greg Cairns, summarizing the 4 phases: absorption, distribution, metabolism, and elimination of drug dispositions. In children, factors that impact drug disposition include physiological changes, impact of the disease and associated therapy, impact of genetics, pharmacogenetics, and impact of environmental factors and nutrition. Second, there is also a need for rational dose selection closely linked to growth and maturation. 2 different approaches to dose selection exist, namely the top-down approach based on population pharmacokinetic models and a bottom-up approach based on physiologically based pharmacokinetic models. The principle of these models are to incorporate genetic, physiological, demographic, and clinical covariates of patient populations in order to predict drug disposition. Their use in pediatric population need to incorporate additional information on early physiological development, such as age-related changes in protein binding, ontogeny of specific cytochrome P450 enzyme or transporters, or markers or renal function. Bridging techniques may use informations obtained in animals and even juvenile animals, adult data, and data in all the pediatric age group, along with in vitro data. And in the example presented here, we were able to demonstrate with fluconazole an agreement in clearance values between real data obtained in neonates and neonatal values extrapolated from in vitro, juvenile, and adult data. Preclinical innovation may also concern better selection of juvenile animal models, such as the mini pig, as human pediatric surrogate for clinical pharmacokinetics and pharmacodynamic drug testing, or the juvenile rabbit to mimic the characteristic of neonatal bronchopulmonary dysplasia. This model is the smallest model that include a factor of prematurity, and it seems a promising model to explore bronchopulmonary dysplasia a frequent and disabling consequence of preterm birth model. Or the juvenile rabbit. In terms of efficacy and safety, there is a need to identify new efficacy markers, new safety markers, target organs in toxicity studies, and also to validate adapted pharmacological tools and use these data from non-clinical studies to optimize the early phases of development. Promising techniques also include organ or tissue engineering, using induced pluripotent stem cells that are derived from animal or human cells obtained from healthy subjects or from patients. This system, based on living cells, allows to explore major function of a specific organ or create in vitro disease models. Placenta on a chip is one of the examples. However, these techniques are far from being currently used and need to be expanded in the developmental pharmacology domain. Additional innovation includes the application of medical nanotechnology in various areas: drug, medicine, therapeutics, in diagnosis of disease or abnormal conditions. The promising of omic sciences are important to characterize individual phenotype based on genomics, transcriptomics, proteomics, and metabolomics for a multidisciplinary understanding of biology, disease pathology, and response to treatment. And there is also a need for very powerful bioinformatics to integrate and analyze all information related to the environment, to pharmacokinetics and dynamics, and to health data. In conclusion, the last challenge is the translation to clinical practice and adherence of clinicians to the new findings. This is easily illustrated by the difficult implementation of pharmacokinetic models in drug evaluation and clinical practice, by the limited use of the new neonatal biomarkers in neonatal clinical trial design, or by limited implementation of pharmacogenomic biomarkers in therapeutic drug monitoring. While difficulties do exist, translational medicine remains an active and rapidly evolving research area, and EPTRI contributes to stimulate this translational medicine in pediatrics. Thank you.